Now that we have a basic understanding of all of the reagents that are possible for EAS and how a single substituent can affect the overall substitution, now what we want to do is go through and see what could happen or how it adjusts. So one of the things that I will kind of step back from is first let's go through and simplify this and say that we were just given toluene and bromine. And if we assume single substitution, if we go through and look at our substitution patterns, we have the potential for ortho, for meta, and for those of you screaming, but just hold your breath. Actually, don't hold your breath because I don't know how long it's going to take to draw everything out. Or para. So we have those three as potential options. As an immediate thing that we could go through and do is if we spent some time having memorized that atom X, so if we go back and look at it, this is our atom X, and that atom X has more electrons than a hydrogen. So that means that this is an activator, and that this should direct, if we've memorized this the way I've suggested, para-ortho. Okay? So that means meta doesn't make any sense. We shouldn't be getting any of the meta product. Between the para and the ortho, which should we get? Well, if we've memorized that process appropriately, we said para first. And so we should get para as the predominant product, followed by the ortho. And the reason we get that para is that steric kind of repulsion. In this case, CH3 is relatively small. We're probably not going to see a huge favoring, maybe 60-40, but we still want to kind of shift towards saying it's the para. The reason for that, we could go back and look at the resonance structures. Oh, goodness. Computer check. Good. We could go back and look at the resonance structures as we had initially analyzed, or what we could go through and do is run the reaction first and see what we get for our tetrahedral intermediates, which is the official response. This is what you should be looking at, is those tetrahedral intermediates, because those things exist. And if we draw out all of our resonance structures for those, what we'll end up seeing is that this resonance structure that I just drew is a special one because we have a tertiary carbocation. And that tertiary carbocation is much more stable than the secondary carbocations. And if we went through and did the exact same process through our ortho and through our meta, ortho we'd note we would also get this tertiary. The meta won't get any, okay? Or won't get any of that tertiary stabilization. And because there's not that secondary thing to stabilize the overall intermediate, we don't see the meta configuration. So now we've got all of those pieces kind of whirlwinded within 2.5 minutes, I guess, depending on how fast you want to say when I started talking about. What we're now doing is taking this a step further and saying, well, what happens if we have yet another substituent on there? Because when we had one, we could just say, well, how does that one atom X affect this? Well, now we have a new dilemma. That new dilemma is because we have two atom X's. Okay, we've got this carbon and we have nitrogen. Okay, so maybe it's not a big deal. How would they go through and direct? Okay, well, the carbon has more electrons than hydrogen, so we would expect para-ortho. And we'll color code these here. So para or ortho. Whoa, so I throw my pen around. The nitrogen, we might say initially that yes, it has more electrons than hydrogen, but we have to remember the Lewis structure. And if we look at the Lewis structure for that, the nitro actually puts a positive charge on the ring. It is not more electrons than hydrogen, is actually less electrons than hydrogen. Okay, so no more but because we've got the Lewis structure to support it. What does that end up meaning? It would direct to the meta position, and we would get that kind of overlap. So now we have meta to the nitro would mean here and where that methyl is. Right? So clearly where the methyl is, we're not going to get a substitution because the methyl is there. Remember, these substitutions are for hydrogen. So if we looked at it just at that analysis, all 4 positions could potentially react. Well, is there something that we could be doing to limit how we evaluate it? Well, remember, the meta director was a deactivator, which makes it less likely to react. The para-ortho was an activator, and it more likely to react. What does that mean? Well, that means that that blue position, the nitro, is actually, all it's doing is slowing the reaction. It's not stopping it from occurring, and it's definitely not directing it. So we're looking at the ortho and para positions to the CH3 being our most active. So the next layer, how should it direct? Okay, where should we actually do that substitution? And a lot of that's going to be a bit tricky, and we'll probably get a mix of potential products. But this is part of our kind of reasoning through this. So first and foremost, if we clean all of that out, let's go through and label each of those positions as different colors. Okay, so we have red, blue, and purple as potential products. Where would we expect our reaction to occur? And perhaps more importantly, why? And before you get too overexcited about this, I also picked ones that would be challenging, right? That we would likely be expected to see a mixture of products, because I don't want a definitive answer per se. I want you to be able to think through and reason each aspect. Oof. So we've got our 4 products now, at least close enough to them. Hopefully, I would argue that this blue one makes no sense, or not necessarily no sense, but makes the least sense. Right? We would get a steric repulsion, which we'll call that S, between the nitro and that Br. We would get that same steric repulsion here. We would get a steric repulsion between the bromine and the methyl that we wouldn't see in the red, but we would also see in the blue. So that blue structure really has 2 steric things pushing against that being a valid product, right? Or a likely product. If we then move through to the red versus the purple, it does become a little bit more challenging to nuance this because we're trying to decide which is larger, CH3 versus nitro. And there are tables of kind of size that you could look up to see that preference. In this case, I would argue your nitro is probably bigger because we're looking at nitrogen and oxygen having more electrons than our carbon and hydrogen. Those bonds are probably going to be a little bit more spread out, and so the nitro is probably bigger, but it's going to be tight. Okay, and so we would ultimately look at references to decide between which of these. On an exam, I wouldn't give you both because that is a challenging thing to nuance. Okay? Or if I gave you both, I would ask why. Okay? So you would just go through, if you wanted to say the red one was in preference, your answer for the red one is because the nitro is smaller than methyl. If you wanted to say the purple one, you would say the methyl is smaller than the nitro. Okay, so it's that kind of analysis that you should be thinking about when you're applying this content. None of you are going to become synthetic chemists for the most part, and so I don't care about the absolute correctness, because that absolute correctness is going to require a little bit more content than we have, so don't stress on that. Stress about what is the logic that you're using to achieve that answer. If we move to the next one down, okay, what happens there? And if we went through and evaluated that similarly as we did the last one, hopefully what we'd note is we'd get the same kind of system. We got that carbon again, carbon with more electrons than hydrogen, so para-ortho. The para is blocked, so we would go to the ortho positions. Okay, and because we needed it last time, we'll throw in that activator. The atom X on the top case, the oxygen definitely has more electrons than hydrogen. We aren't looking at any kind of weird resonance in that case. This is going to be para-ortho and activator. We run into a similar issue here, and that all 4 positions are again activated. Okay? How could we go through and decide this? In the first question that we looked at, we had a clear difference in activating ability. We had an activator versus a deactivator. In this second case, they're both activators. So how would we decide which one? Well, is there a difference in their activity? Oxygen is much more activating than the carbon because the oxygen has those lone pair electrons, and all we need is one pair that can actually donate in. That means the oxygen is a stronger activator than the carbon, which would then suggest the para positions would be our most likely positions. Those are identical due to symmetry within the molecule. And that means we would expect the nitration to happen para to the alcohol as opposed to the methanol or the methyl. Okay. And so again, what you're trying to reason through, this one's a lot nicer because we don't have to worry about size or secondary steric effects. We're just saying explicitly that's what's preferenced. Okay. So hopefully that makes sense and you're starting to kind of put together these pieces. We're looking at electronic effects, which some of which can be memorized, that para-ortho activating or deactivating aspect, and some of which is sterics, and that sterics is a little bit more challenging to nuance, particularly in these smaller cases. So if we were going to push to something that really required sterics, I'm going to throw on a large steric structure, kind of like, oh, never mind. So our lessons, activators trump deactivators, right? Stronger activators trump weaker activators. And if we consider this really as kind of a scale, it's really just one rule. That one rule is that we have activators and we have deactivators on a scale, okay, with markings all the way across. The further you are on the activator side, the more likely you are to dominate or control the reaction. Okay? So when we compared the activator to the activator won. When we compared the activator to a slightly weaker activator, the stronger activator won. Okay? So now to that steric argument, if we go through and evaluate both of these cases, both of them, okay, in each system, we're looking at carbons. Okay? So there is no difference in activating or deactivating ability. There's really just atom X and it's a carbon. Okay? Because it is just a carbon, that means it's going to push as an activator para-ortho. So again, what we should go through and do is come up with a labeling system to see how it would direct. The para for the top one is blocked by that methyl, so there's no hydrogen to substitute. So we'd label those as our ortho. Okay, whoops. For our carbon, our other carbon, again, the para is blocked, so we get all 4 being potential reactive sites. We could eliminate 2 of them because of symmetry, again, symmetry through the molecule. I don't think I like that black line on there. If we do the same thing down below, para, note this time we have the ability to do all of them. And then for the methyl, ooh, interesting. Okay, so if we go through and address the top one, our top reaction, we would again expect 2 potential products. We'll call it the red one with a nitro. And I think I drew the double bonds a little bit weird, but that's okay. Remember, resonance, those double bonds can be anywhere within that ring. And then we could have the blue product with the nitro. So how are we going to decide this? This is solely that steric argument. We get a steric repulsion here, and we get a steric repulsion here. Which one is a larger steric repulsion? Well, which one's bigger, a methyl or 3 carbons, okay, an isopropyl? The isopropyl is certainly larger. That steric repulsion is going to prevent the nitro from reacting, which means that's not the product. We expect the blue one to win. Oops, and I called that a nitro. That was an isopropyl. Okay. What happens when we move to the bottom one? Well, that one becomes a whole lot trickier because the same 3 positions are activated. So where would we expect the product to form? Okay, so we can call it purple, green, and we'll throw in an orange. So if we were again to show each of our products, there's our orange. Bothers me that I drew it that way, but that's okay. Our purple. Okay, and I believe I saw a post something about, hexagon drawing class. This is the unit that people reference when they're talking about drawing too many hexagons. If you hadn't guessed that yet, you just have to draw a lot. So we really have those 3 potential products. Note that bottom position had no directing effect associated with it, so no substitution will happen there. So again, we would go through and kind of evaluate. There's a steric repulsion. There's that same steric repulsion. We move to the end. I guess I can use a blue. We get a steric repulsion here, and we would get that same steric repulsion there. So what does that ultimately mean? Well, purple has 2 steric effects against it, so purple is bad. If we compare the orange to the green, the orange one has that black repulsion if we're color coding it, and the green one has that blue. Okay. What we're again comparing is the size of the isopropyl to the methyl. The isopropyl being larger, we would expect a larger steric effect in the orange structure. That large steric effect is going to prevent the nitro from reacting at that position, which would suggest the orange one is not our product, and we end with the green in the highest yield. Okay, so that's the process of moving through these. Hopefully that makes sense. It's about to all get deleted here in a second. If you don't want it, hit pause. And that's what you should be thinking through when you're looking at these multiple substitutions. Where this is going to next move is that this particular unit, I think, is one of the easiest ones to test on synthesis, because you're looking at relatively few reagents, right? And you don't have to worry about all the different things that they do, because they really just do one thing. So if you memorize those reagents, there isn't a whole lot of questions that could be asked. You just, oh, well, that means it puts on a nitro, like that's it. So where this becomes more challenging, or where the level of content kind of rises naturally as far as questions go, is now synthesis. Given a benzene, how do I end with this other benzene structure that has 3 different things on it? Okay, because the order in which we put those things on is going to change how the next one can be put on. So it really, really lends itself to that synthesis object. So if we take a look at this, we've got 2 things on here. So this is the last thing we're going to do kind of before synthesis. It's along those lines. Okay? And if I went through to try and run this reaction, what I might just go through and say was the only thing that changed was a bromine. To get that bromine on there, I need Br2, FeBr3 is my catalyst. Okay? Well, atom X is a carbon, and it directs para-ortho. The para position is available, and so are the orthos. Well, if that's the case, the product that I would expect to get is the para, not the ortho. So how can I ensure that I get the ortho substitution? Okay? And that is a challenging question, and we would have to go back to our reagents. And one of our reagents, in fact, technically a couple of them, but one more easily than the others, had kind of a cool feature. Okay? That cool feature was that it was reversible. we could go through and effectively do a protecting group. We could run some kind of reaction where we put on a protecting group here. Then when we go through and run the reaction with our Br2 and FeBr3, just trust me, that says FeBr3. Now we could put our Br3 on, or our bromine, sorry, and then we would deprotect to get that final answer. So we would have to know, what is that protecting group? What was the reagent that allowed us to both protect, or not protect, but put something on and then immediately take it off? That was H2SO4, concentrated H2SO4. And the deprotect, which honestly I should have looked up just to make sure, but the deprotect is also H2SO4, except I think here we specify aqueous, okay, or slightly more dilute than the concentrated. And that reverses that reaction. Okay, so now what we do is we've put on that sulfate. So it's no longer just a protecting group here. We would put on the SO3. That SO3 blocks that position, the Paris position to substitution, so that when I do the Br2 and FeBr3, the only available site is at the ortho. Okay? So you might say, well, that's sterically hindered. It is sterically hindered. That's why we would probably have to push on this with a little bit of heat and probably time to allow that bromine to react to that spot. Once we felt we had a high enough yield, then we would go through, isolate, deprotect, and we would have our final product with the bromine at the ortho position. Okay, and so that kind of protecting can kind of happen. It's not really protecting per se, because you are changing the structure quite significantly, and then just reverting backwards. Okay, so in this case they call it a blocking group, not a protecting group. 6 of one, half dozen of the other, I think is the statement. So then the last thing I kind of want to address here would be our true kind of synthesis. In this true synthesis, what I want to start with is benzene. And how do I get it to become that product? Okay, so the first thing that I would go through and do is try to identify how those pieces came about. Where did those things come from? Well, the red one, hopefully we recognize, is the Friedel-Crafts alkylation. Make sure I got the carbons right. 1, 2, 3. I think I did it. Okay, and aluminum trichloride. The blue one would be Br2 and FeBr3 we just showed. And then the green one would be nitric acid and H2SO4. And for those of you saying, well, if I knew those, wouldn't I know the answer? Well, which one goes first? Should it be 1, 2, 3? Should it be 1, 2, 3? Now, since I'm in green, 3, 1, 2, 3, 2, 1. Hopefully you get the idea that we could come up with multiple different variations on how this could come together, and you're going to be responsible for acknowledging what that would mean. Okay? I find the easiest approach to this is to look at the arrangement of those groups and see if you can call out some kind of directing effects. Make sure I color code these right. So the green one as a nitro is electron withdrawing, so that would be a meta director. And if we look at the substitution, it might have the acyl group meta, reasonable, but then it doesn't have the bromine meta. Okay, so I would argue the nitro is probably not the best bet to go first, okay, because the arrangement of those groups isn't there. So I'm also not positive I put in the proper sequence. If we look at the red one is that carbon which again we might say has more electrons than hydrogen, but remember that resonance, it has less electrons. It's also a meta director. And if we go, yep, it's meta to the nitro, fantastic, but then it's para to the bromine, which doesn't work. That would suggest that I should be doing the bromine first. Okay, so there's one where I have the bromine first. So let's run that one out just as written and see if it worked. Okay, so the first thing I do is the bromine. Okay, well, actually, let's run out the other option. It could have been 2, 3, 1, right? Hopefully everybody sees those patterns within it. So 2 seems to make the most sense. If we do that, what happens with our alignment? Bromine, remember, is a weird one. It is deactivating, but it is para-ortho directing. We're para with that, and we're ortho with our nitro. So that would seem to be a good option. So now the next bet, what should we do next? I will remember para-ortho, which means it's going to direct para preferentially. If I did the nitro next, that's probably going to place the nitro in the para position, not the ortho position. So that probably means that step 3 shouldn't happen right away. I probably want to do the acyl group first. So to do the acyl, just standard, we run that reagent. Okay, because it's an acyl, it doesn't need to be an activated ring, so that's good. That would suggest going 2, 1 makes sense, or blue, red. And then I could go through and throw on the nitro by adding the nitric acid, and sure enough, that's going to be our best bet. Okay, so when you see these, draw what the reagents are. Okay, and if you have trouble with the reagents, look at your multiple choice answers. Okay, see what the multiple choice answers are, because they're probably just permutations, as we just drew out, of the same reagents. So now hopefully you'd be able to identify that that red one very clearly belonged to the acyl, the blue one that we drew out very clearly belongs to the bromine, because those are the only atoms that are really associated within those reagent sequences. Okay. It again does take a little bit of practice, and there are situations where you run into problems like the acyl group, or not the acyl, the alkyl groups. Remember, Friedel-Crafts alkylation has a couple complicated issues. If you need help remembering those, you have to watch it in actually 2 videos, and you might be able to get away with 1 video, and that was that last video that you probably just watched. Okay, with that, we've covered, the end of this video. The next sequence of stuff, we start seeing some kind of weird things happening with aromatic rings. I think they're kind of cool, but they are a bit bizarre at the same time. And so with that, thanks for watching.